We'll see you.
No, I'm just setting it up. Is there? Oh, yes, they do have chargers. All the text is backwards, though, so that'll be fun for people.
I could probably put this closer than these.
Put it closer.
So that'd be better for the microphone. Moving the chairs. Oh, you know what would be better?
What?
Over here.
What about things here on like wide angles?
Fish eye.
Okay, I'm clicking on the link and seeing if it's working. Nice. Not sure.
Here, it's too close. It's like where I want to
Go
put
back where the computer is.
Oh, I need, yeah.
The purple one? Would you just need like a little notebook or something? Okay. She's meant for me. Hi. Thank you. I feel better that I practiced this morning, so it's like fresh in my head. I think it's going to be too long, for sure. But I didn't have time. Yeah. I don't think they're going to I've never seen that. I'm just going to tell them it's too long. And it is what it is. I did the best if it's 6 years. I don't have her. It may not be more. I think I just need to spend less, a little bit less. Everything. Or I think I just describe. Yeah. On the page.
Natalie, the settings on this camera?
Consumed by PhD life.
Hmm.
One day. Well, yeah. Right around.
Hey, Natalie, the setting for this camera?
A little, I think any of them really.
I have it on, just on AV.
Okay, and then what you do is you switch this.
I already did.
Okay, and then to start recording, you hit this like start button, and then it starts recording. See?
Okay.
So now you hit it off. Okay, now it's not recording anymore.
Okay.
So after 30 minutes, that will go off.
So do I want?
Do you want me to split some of the chairs?
Yeah. Particularly that one.
Hey, Connie. Good morning. Are you excited for today? I'm excited for today to be over. More specifically, hey, Steve. Good to see you. Okay, I thought it was late, so I don't know. Okay. Which chair, Steve?
This one right here. No, second row.
Okay. This one?
Yes.
Is that good?
Mm-hmm.
Steve, let me introduce you to a couple of people. This is John, my husband.
Yes.
Yeah, Steve. Yeah, it was way back when I first started, and now retired, and now still teaching as an adjunct professor. And helping Dad?
Yeah.
Cool.
Doing some miscellaneous things.
That's awesome. Are you gonna teach your own class eventually? Or?
We're just talking about letting have the class.
And then back here, we did already, actually. Yeah. So this is my father-in-law, Jim, and my father, Jack. And this is Steve. So he was an engineer with 9 Springs wastewater treatment plant that helped fund my research. Way back when I first started, he taught me all about aeration and diffusion of oxygen and then now retired and now teaching here. Yeah. It's good to see like people. I've been like and now seeing people are like,
Already has.
yeah, so that was kind of crazy, trying to move and shit. So yeah, that was no, I got early so that you're like, it's just, it's too long. But it's like, I think I talk about things too much. Maybe your first life
Well, maybe more so they can say now. You know, now I'll have like 2 days. Really bad and don't know. Oh, so it's 4 days, so he gets back. So they have like.
Okay.
Oh, yeah. Yeah, it just took a breeze through it.
Okay.
Natalie.
How are you?
Good, how are you?
Good. So this is my husband, John.
Nice to meet you.
This is the guy that did all the F&R stuff.
Oh,
For me.
FNR stuff.
And he's the one that recommended my paper so fast.
Oh, okay, yeah.
Yeah,
Just
yeah.
like those.
Yeah, no, they did. Yeah, no. That was helpful. I made the changes that you made, and I'm sure we'll go through more.
Oh, yeah.
For sure.
Yeah.
I did not, I don't know, I was gonna ask Dan, because we're gonna talk about everything after the session. And since you're involved, it might be helpful to talk to Dan. Yeah. I mean, I think it would be in my presentation. Okay. I'll ask Dan, because I don't know. Oh, you got me my clothes. I got a hole in the screen.
Yeah.
Are you sure that's gonna be faster?
Yeah, I will.
On stage today.
Yeah, I guess. Yeah, so that's
2 minutes?
why. I want to go to the design. Not even looking at them.
Here. You sure that's faster? Long time no see. How are you? We're fixing up work.
Better not go there. Very, how are you?
I was kind of laughing, so I'm like, I'm in the, I got it. I'm like, that's gonna be so unusual.
Yeah.
Yeah.
I'm always an engineer father. I started doing that like for a couple of weeks, then I realized it made more sense I was and everything. Okay, when you yeah, sometimes I go like early in the morning or later, but during the daytime, yeah, I think in the morning, usually doing that. Yeah, so that's why we just. Cool. I moved to Milton yesterday. Yeah, so next time we go to Costco. Yeah. Yeah, now the children stay. Yeah. These days on that, and then I review a paper journals that's due this week. So yeah.
This is my dad, my mom.
Oh, they're all cookies.
Yeah, he told me it was just Alex is
We're not crashing. Thank you. Yes.
Is that a bot?
So really makes me look really weird.
Okay, is it near? Yeah, so I'll show you. No, it happens just like super fast. It takes 30 minutes, 37 minutes to be here. Oh, yeah, it is. This is the company I work with. This is where I live. And this is Costco.
Okay, so it's near, that's near the station.
Yeah.
Oh, I see.
The market.
Yeah.
There's no box. That's yeah.
Hello, ladies. How are you?
Okay.
So have you're now buttermilk, right? Developing
It's just all that. Usually has it's so sorry. But
I might be able to move a little bit too. Let me actually move this.
probably in the middle, so it's probably better for you.
Well, I might position it a little bit this way just because of where Greg is sitting. I think that's great. That'll be good. This will be fine. Thank you.
You can test your video grades.
Was this you? Oh, sorry. Great.
I brought my old reminders. Nobody wants to sit in the front row. It's not a pretty thorough intro. No, yeah, I don't have the comfort of this. I think Garrett, I'll try to, you know, regulate it, but, what's your 11? Okay, I'll try to keep my comments really long. Matt Scarborough, Tommy Poncho.
Natalie, is this position good?
Yeah.
Okay.
Well, thank you all for being here. Very exciting time to see Natalie's final presentation here. Natalie came in 2013 from Colorado, bachelor's degree in environmental engineering. And she joined us when we were starting a very interesting project of taking oxygen that we fit into wastewater treatment plant and bringing it down slowly to improve the energy efficiency of wastewater treatment plants. And at that point, we had an idea. We had done a little bit of work in the laboratory, a small reactor, and we were set. The Madison Metropolitan Sewerage District came to us and said, "We know what you guys are doing. Can we try something at the pilot scale?" But sure. And then Natalie came as the person who was going to do this. The pilot plant has been in existence for about 25, 30 years. And last time we had operated was about 10 years ago. So she had the responsibility of looking at a lot of big tanks, changing everything. She was going crazy trying to figure out how to not get the water not to leak from there. My favorite picture, which I don't have, but she says she has it in there. She's inside of the clarifier. This is a big tank that we use to separate the water from the bacteria, and she's inside trying to fix things. She has been an excellent person to have in the lab. She has the I can do attitude all the time. She's been the teaching assistant for many of my classes. I started teaching online. I can do it. There was Natalie. My house got flooded. I can do it. Got flooded at the same time that we did. But we have experienced a lot of things, and I gotta say it's this generation of people who are graduating now, it's been excellent to have, and Natalie is, I thought it was the last, but I see somebody that posted some papers here and graduated. Yeah, So it's been a real pleasure to have them all here, especially Natalie, and she's going to tell us about what she did with this low oxygen idea. So we went from pilot plant to other things, trying to figure out what these microbes are and so on. So I'm looking forward to it. I haven't read the last chapter of her thesis, so we'll see how that goes.
So yeah, like Dan said, I've learned a lot while I was here. I learned way more than I expected, to everything from electrical work to plumbing. And then I also learned a lot about myself. One of those things being that I am terrible at estimating how long things take, one. So that's why Dan hasn't read my chapter. And two, I like to talk a lot. So if this presentation is a bit long, yeah, just know that it's because I'm really passionate about what I do, and so I apologize in advance for the length of it. So like Dan said, we wanted to investigate biological nutrient removal using low oxygen, and I'll explain all of how that all ties together during this talk. So like Dan said about me, I came from Colorado. I got my bachelor's degree from Colorado State University. That's where I grew up. And then I went to work at Northern Colorado Water Conservancy District in Colorado. I was there for 3 years as an intern, and I worked there full time in between undergrad and grad school because I graduated in December. And I learned a lot about water resources, but I knew that my passion was with biology and microbiology, and so when Dan had a wastewater project, that was a no-brainer for me to take it. So I started off with my master's. I was like, I'm not doing a PhD, I'm only doing a master's. And then in I don't know when Dan started bugging me, but Dan started bugging me about staying, and I was like, no, I need to work. I need to go work, pay off these student loans. But you know, then Dan, my favorite story is when my favorite story is when Dan's like, okay, Natalie, well, if you can't make this decision, then you know, how about I'm going to bring on a new student for you to give a tour of the pilot. They're going to take over the pilot plant for you. And I was like, wait, Dan, I was actually going to talk to you about it. I'm going to stay. That's what I decided. So I ended up staying for an additional, 4 years? Time flies. So I've been here for 6 years now, working with Dan. And Dan, I just thank you for giving me this opportunity. I'm trying not to cry right now, but thank you for believing in me and for giving me this opportunity to, even though it was PhD is a process, and an exhausting one at that. But I've learned so much, and I attribute that to you, and you've been a great mentor and teacher, and I'm so grateful for you. So I have to start off by saying that all of this work was not done by just me. I had the opportunity to mentor 14 undergraduate students and high school students during my time here. And that was one of my favorite parts of this, was getting to work directly with these students, teaching them what I know, teaching them how to run a reactor, teaching them how to troubleshoot, teaching them how to be proactive. And a lot of them have become my close friends. So I have to start off by thanking them for all of their hard work in keeping these reactors going. Because like I said during my master's defense, taking care of bioreactors is like taking care of a kid. You take care of it every day or every other day, which Dan thought was great advice for new parents. All right, so now we're gonna get started. So the bigger picture. So I look at nutrients, nitrogen and phosphorus, but this talk I'm gonna focus on nitrogen because it's the common theme among the 3 projects that I worked on. So why is the nitrogen cycle so important? Well, the National Academy of Engineers came up with 14 engineering grand challenges for our future as a population, and one of those 14 is managing the nitrogen cycle. And I really like this quote where it says, it doesn't offer a catchy label as global warming, but human changes to this nitrogen cycle has dramatic environmental impacts that we all need to, we should care about, and it's part of the reason why we do the research that we do. So, that's a special effect. The Earth's atmosphere, to begin, is made up of 78% nitrogen gas, or N2. But this nitrogen gas is tightly bound by this triple bond. And it takes the energy of lightning to break that bond, or specialized microorganisms that can break that bond and turn it into a nitrogen compound that is usable by living organisms. Nitrogen is part of our DNA. It's part of our, it's part of amino acids. It's essential for life. But unfortunately, I guess I'll hold on. One more slide. But until recent times, nitrogen fixation, or this little arrow right here with the lightning strike, was performed, like I said, very small amounts by lightning, mostly by microorganisms. And that was until the Industrial Revolution, when the Haber-Bosch process was invented. And what this process does is it takes nitrogen from the atmosphere that's highly abundant, is a reservoir of nitrogen, and it turns it into something usable, like ammonia at the end there in the yellow tank. And so that's usable as fertilizer for crops. So this has been an essential invention for modern crop production, so that we can, have a food supply for our growing population. But the side effects of this were something that maybe they didn't understand at the time. So essentially, humans have double the rate at which nitrogen is removed from the air, and it's introduced into the environment, into these biologically available compounds. And so with the widespread use of these fertilizers, of which many times only half of it is taken up by the crops and the rest leaks out into the environment, it leads to eutrophication, like this, which we see often in Madison lakes, which is the over-fertilization of these lakes by these nutrients that leak out. And essentially, the algae blooms, these algae blooms, they grow, they die, and oxygen is consumed from the water. And these aquatic life, fish and other aquatic life, require oxygen to live. So this is detrimental to this aquatic life. And not only does it affect these freshwater bodies, but coastal regions are also affected. So when nitrogen leaks out into rivers, it travels to places like the Gulf of Mexico. And there, you can see in step 1, the nutrient-rich water flows in. Step 2, the algae grow, feed, and die. Zooplankton then eat the algae, breathe. 4, bacteria feed on this fecal pellets and dead algae, and they consume oxygen in this process. And then 5, that's where they deplete the oxygen. And 6, lastly, the marine life they either have to flee to another environment that has oxygen, or they die. And for over a century, this is what we understood about the nitrogen cycle. And so why this is important is because the more we can understand about these processes, the more it relates to these global issues as well. So here we have what we have understood about the nitrogen cycle for over a century, where we start off with the conversion of organic nitrogen into ammonia. Ammonia is then oxidized to nitrite by the red arrow. Oxidation is going up. That means that it requires oxygen for that process to occur. And then nitrite is oxidized to nitrate, which is then denitrified back to nitrite, and then denitrified to nitrogen gas, which then goes up into the atmosphere. Now, like I said, the red process requires oxygen, And the blue process, the reduction, requires organic carbon for that to occur. And I made little figures to represent, nitrous ammonius, nitrospira is a nitrite oxidizing bacteria, nitrous ammonius is an ammonia oxidizing bacteria that takes ammonia to nitrite. And then an example of a denitrifier is chorobian, and that one will come up later in this presentation, but there are many organisms that have the ability to denitrify. Now, over the past 3 decades, scientists have redefined our understanding of the nitrogen cycle. And a lot of this has come through genome-enabled research. So if we look at this plot, this shows the cost of sequencing a genome and the year, and as we advance into the future, it becomes cheaper to sequence these genomes, and that makes this more available to researchers like us to look at not only who the microorganisms are in certain systems, but how are they functioning, what are their capabilities, and what are some of the things that may lead to their adaptation to certain environments. So engineers love to use this black box where we look at, what's coming in and what's going out, but we don't necessarily know what's going on inside of that box. And this too has been valuable because the observation of changes like this has led to discoveries of certain microorganisms, and then genome-enabled research has taken that even further to help us understand what's going on. So far, like I said, over the past 3 decades, there have been many discoveries. So one of those is Anammox, or an anaerobic ammonium oxidizing bacteria, which is unique because it can oxidize the ammonia, but it doesn't need oxygen to do that. And it also does not need organic carbon, like I mentioned previously. And Anammox is unique because it was one of those examples where it was discovered in a wastewater treatment plant, because they were observing this process was taking place, and they were seeing this ammonia going away without any oxygen or organic carbon, and they're like, what's going on here? And then genome-enabled sequencing or genome-enabled research has allowed us to further our understanding of these microorganisms. We've also discovered that ammonia oxidizing organisms are not just bacteria, but also archaea. And more recently, in 2015, since I've been here, there was an organism called Comomox that was discovered in some bioreactors in Europe. And this was pretty exciting because it's always been believed that the process of nitrification here, from ammonia to nitrite and nitrite to nitrate, occurred through 2 organisms, 2 distinct organisms, ammonia oxidizers and nitrite oxidizers. But they discovered that there was actually one organism with the capability of doing both of these processes, which was pretty mind-blowing, and I'll talk more about that organism later. And then Anammox, what you can see here is how all of this comes together and has changed our understanding from this as all the nitrogen transformations to actually this is what's going on. And this is what our understanding is now, that there are 15 transformations and 8 different oxidation states of this inorganic nitrogen. Anammox, we'll talk about more about later, but they carry out the process 10 and 11, so they go directly to nitrogen gas, and it's viewed as a shortcut to this nitrogen cycle, and engineers have utilized this to improve on biological wastewater treatment processes. So continuing with the bigger picture, but kind of narrowing down a little bit in the wastewater, there's a new paradigm, what we call, for the water resource recovery facility. So it's no longer just wastewater treatment. It's now, what can we recover from this water? Well, we can recover nutrients, energy, and water for reuse. So the Nine Springs Wastewater or Water Resource Recovery Facility here in Madison, Wisconsin is interested in this new paradigm, and they're very interested in becoming not only energy neutral, but energy positive, potentially, in the future. So that's where they generate more energy than what they consume. However, If we look at these basins here and over here, that is where the biological nutrient removal takes place. And in these basins, you'll see that there are, I don't know how many, I would estimate thousands of these air diffusers, which are similar to what you put in your fish tank to provide oxygen to your fish. We also need to provide oxygen for some of these bacteria that carry out the nutrient removal process. As you can imagine, all of this takes a lot of energy. And for 9 Springs, it accounts for 40% of their total electricity requirement. And in some plants, it's up to 60%. So, if we're going to work towards energy independence, we not only need to look at, well, the biggest energy consumer, this oxygen, but then also decoupling their carbon and nitrogen demand. So, if we look at the waste coming in, there's organic carbon and nitrogen. Organic carbon can be used to make biogas, like methane, for heat and energy production for the plant. And then nitrogen removal requires energy and carbon, but we're interested in trying to split this requirement so that we can generate as much energy as possible from the available carbon, but also remove nitrogen and achieve the treatment requirements that we need to achieve in order to protect the environment. Okay, so here is an overview of a little schematic that I made of the 9 Springs Water Resource Recovery Facility. So we'll go into more detail about all of this because you can see there's a lot going on. If you start at the top left, that's where your influent wastewater comes in from the collection system. And then it goes through these primary clarifiers, which allow the water to separate the heavier materials, the solids, from the clear water, which just contains dissolved compounds. And then the water with just the dissolved compounds goes into the biological nutrient removal process, and then goes into the final clarifiers where we settle out all the microbes and let the water go to disinfection and then the environment. The focus of my first chapter is on this biological nutrient removal process here, which we call a mainstream process. And so, now I get into what I've been up to for the last 6 years. So, chapter 1 of my dissertation is on total nutrient removal with low oxygen at pilot scale. Low oxygen being one strategy that we can use to reduce the energy and electricity requirements of a wastewater treatment plant or water resource recovery facility. 2, that new organism that I told you that was discovered in 2015. My second chapter is on detecting them. And do we find it in low oxygen systems or in high oxygen systems? And then lastly, I look at a separate system that uses anammox to treat water and look at the key microorganisms involved and their activity. So we'll focus on the first one. So we'll go back to the biological nutrient removal process. For some of you that may not be familiar with this, this is what the schematic looks like. There's many different configurations of this, but this is what the 9 Springs treatment process looks like. So again, water comes in, and then there's different zones. So these different zones are different environments that we can control in order to control how these microorganisms work. And so you can see from the red line that as soon as this should be shifted a little bit, but as soon as the ammonia gets to the aerobic zone, then the microbes can use the oxygen to convert the ammonia to something less toxic, like nitrate. Now, plants that have a total nitrogen removal or a total nitrogen removing requirement, which some plants still don't yet, but that's something that is being anticipated as a regulation that's coming in the future. They also do denitrification. So the water is recycled back to the anoxic zone, where the nitrate is then reduced to nitrogen gas by taking up organic carbon. One problem with this is that some plants do not have enough organic carbon to achieve this total nitrogen removal. And then lastly, I want to point out that in the upper right-hand corner, that is how these microbes grow in this system. So they grow in something called a microbial flock that includes filamentous bacteria, all of the bacteria that we care about, along with other bacteria that just grow but are not contributing to the nutrient removal directly in this process. And the reason why these flock are important is because that's how we settle them out in the final clarifier. So they're heavier, so they settle, because we don't want these solids to go out into the environment either. And then again, there's my little microbe drawings. So this process also removes phosphorus and carbon, but again, like I said, I'm focusing on the nitrogen for this one. And then what we wanted to understand and try at the pilot scale, like Dan said, is can we reduce the amount of oxygen that we're pumping into that aerobic zone and still achieve the same level of treatment? So will these microbes still work under these low oxygen conditions, or do they completely change? Like what happens in that process? So, like Dan said, when I got here, I had a bit of a fixer-upper on the tanks, to say the least. So, and I even put my husband to work, John, and he came in and he helped me pull out all this old tubing, and there were pumps missing, and I had to, put new tubing in. First, I had to figure out where to buy tubing like that I needed. And then there were these diffusers that were there, but they were broken, and so I had to figure out how to rebuild it. So I took this stuff home, and luckily my husband's a woodworker, so he has all these tools at my disposal that I could use. There were issues with the clarifier, like Dan said. So Steve Reeser, when we first met, he was like, "Oh, that pipe that goes into that clarifier, the holes are too small and it just clogs and it causes overflow. So that's a problem that we need to fix." So I was like, okay. So I got into the tank because I didn't know how else to get to that pipe. I had to make my new pipe, and my dad actually helped me drill holes into this pipe and figure out how to make it so that it still, when the water flows in, it still lets everything settle. So we finagled something. And then I also had to figure out where to order pumps, and then learn how to take them apart and put them back together. And then finally ended up with a beautiful functioning pilot plant after about 6 months of work. So that was 6 months of my master's, which is getting this thing up and running and not leaking. And then we were ready to take samples. And then we babysat this reactor. I personally babysat the reactor with help of my undergrads for about 3 years before I passed it on to somebody else to babysit. And I was basically on call, which was great. I remember getting some 11 p.m. midnight calls from the operator saying, "Hey, your plant's overflowing." So I had to go and fix it. And then we finally put in some float, like a system in there that would shut off the pumps if the water got too high. So we put the kibosh on all these overflows that were giving me so much anxiety. So again, like I said, some of the main objectives, I forgot to delete that slide. So the main objectives of this project were, one, to identify a strategy to take a high oxygen system to low oxygen. We didn't want to just go low oxygen right away because we've done that in the lab and we saw that it takes a month to regain this removal of ammonia. So we knew that it would work eventually, but we wanted to come up with a strategy that a full-scale system could use to transition to low oxygen without impacting any of their effluent quality, what's going to the environment, because they have to meet requirements for that treatment. Then we wanted to increase our understanding of the microbial community before and after these oxygen changes. What was going on? Did the microorganisms change completely, or were they just better at using low oxygen? Did they adapt? And then finally, we wanted to estimate how much energy would this really save the full-scale plant if they were to do this. And that was something that a colleague of mine, Matt Scarborough, who has extensive consulting engineering experience, he helped me with coming up with a model to estimate how much Nine Springs would save if they were to reduce the oxygen in their tanks. So this is the schematic of my pilot plant. So one of the unique things about my pilot that differentiates it from other systems or other studies is that we took primary effluent from the full-scale plant in real time. So I was experiencing all of these changes in chemical composition and concentration and changes in temperature, just like the full-scale plant was. And then we ran it as a continuous process, so the water would flow in, and then whatever came out, I guess they had a drain that went back and got treated again, just in case, my system wasn't working. So it took about 9 hours for a drop of water to get from the first tank to the last aerobic tank, which is a bit shorter than what the full-scale plant operates as, but that was what we could achieve with, the limitations of what we had, and also the flow rate that we wanted to run at 1 gallon per minute, approximately. So we treated about 1,300 gallons per day, approximately. And to put this into perspective, Nine Springs treats about 40 million gallons per day. So it's quite a bit smaller, but not as small as the little, 3-liter bioreactors that we have in the lab. So the road to low DO, we operated in 3 different stages. The first was at high oxygen, so you can see each one of the aerobic tanks is depicted by each one of those lines in the top graph. And then once we were satisfied with, the level of treatment and the amount of time we were operating at high oxygen, we decided to decrease the oxygen slowly over time. And we made sure to wait 3 weeks of good treatment before we made any changes, because we didn't want to just change it and change it. It takes a while for this microbial community to adapt to all of these changes. And then in the bottom graph, you can ignore the yellow line for now. We'll talk about the pink one. The pink one is our total inorganic nitrogen, total ketone nitrogen, which includes the inorganic and the organic fraction of nitrogen. And then that concentration is in milligrams per liter. And you can see over the course of about 500 days, we were able to achieve relatively good treatment. I think it was about 97% total nitrogen removal as the average. With the exception of these few days, those are when those little peaks there, that's when they called me and told me there was an overflow at midnight, or a pump problem. The yellow line is SND, which is simultaneous nitrification, denitrification. This is something where you have nitrification and denitrification occurring in the same reactor in the presence of this low oxygen environment. And the reason why we cared about tracking that is because, one, it's reported that this occurs at low oxygen, and 2, it's a process that saves the amount of carbon necessary and oxygen, because it's another shortcut in the nitrogen cycle. One thing we were a bit surprised to see is that it was really unstable. It was all over the place, and ultimately, we weren't able to pinpoint why that was exactly. It looks like it went away at the end, but we did operate the reactor for many more days beyond that, and it did come back, but it never reached a stable point. Regardless, we were able to achieve good total nitrogen treatment. We also looked at phosphorus, which I'll talk about very briefly. Overall, phosphorus removal was great. That was actually, I was surprised that when we had upsets in the operation of the system, that phosphorus removal remained relatively stable. It didn't impact those organisms. And then ultimately, we calculated if Nine Springs were to change their maximum oxygen concentration to the lowest point that we achieved, which was about 0.4 milligrams of oxygen per liter. Full scale, to put that into perspective, full scale operates greater than 2 milligrams per liter, sometimes upwards of 4. And saturated air is about 8 to 9 milligrams per liter of oxygen. So we were down to 0.3, 0.4 milligrams of oxygen per liter and had good treatment. And then we calculated that this translates to about $300,000 in savings per year. And that's if all Nine Springs did was just reduce their oxygen. That doesn't include them upgrading their blowers to high efficiency blowers. It doesn't include any other changes other than reducing how much oxygen they're putting into their system. All right, so now to transition into how do we understand the microbial community, what's going on. We first performed some batch kinetic tests. So I actually counted because I think another student asked me to go over the data with them. And I was like, realized there's 100 of these tests that I did because I counted, I actually did 100 of them. Where I brought the bacteria or the sludge from the plant, I trucked it over to the lab, I put it in these little tanks and I controlled how much oxygen I put in. And I was able to set, like, okay, I want to put in 2 milligrams per liter of oxygen and keep it at that level over 4 hours. And then I measured how much the ammonia decreased over time. And from that, I was able to get a rate or how fast they were able to remove the ammonia and plot it like this. So each one of those points represents one of these 4-hour tests. And so what this tells us is how well a population can utilize oxygen. So the blue line with the blue points is the full-scale plant. So when we took biomass from the full-scale plant and we started reducing the oxygen, you can see the rates are lower than that of the red line. And the red line is the low oxygen pilot. So what that tells us is that the ammonia oxidizing population in our reactor was better or more efficient at utilizing the available oxygen in the system. So we call that they have a higher affinity for oxygen. And that can be achieved in a few ways. You know, when we submitted our paper, they were like, oh, did you check this aspect of that higher affinity? Because we were focused on, okay, the microbial community changed, so that could explain it. So if we go back to these flock, one other way that the organisms might be better or have a higher efficiency at utilizing the oxygen is by growing in smaller flock, because the oxygen doesn't have to diffuse into such a big mass. A smaller mass, everybody gets access to the oxygen. And so we looked at not only the whole flock size, but also those microcolonies. So our ammonia oxidizing bacteria, they're my favorite, because when I did microscopy, they were easier to identify because they like to grow in these little clusters or these microcolonies. And so we used fluorescence microscopy to count and measure the surface area of these microcolonies and then compare the full-scale microcolony sizes to the low-GO microcolony sizes. And overall, I think we counted and measured 400 microcolonies and 400 flock sizes. So 800 of these, we took pictures, we measured them, and then we plotted them in these box plots. So at the top, we have the whole flock size, and ultimately, we did see a statistically significant decrease in size. And we also saw a statistically significant decrease in size of the microcolonies. So that means that part of the adaptation to these low oxygen environments may just be in how they grow and are able to access the oxygen to begin with. So then the next thing we did was we decided to profile the DNA of our sample. And one way we can do this is through 16S rRNA sequencing. 16S is a gene that is conserved across bacteria, yet it has variable regions that allow us to identify who is who. And that's the level of resolution that we get from this, but we wanted to kind of get an idea of the microbial community. So we take a bacteria sample, we beat them up, and we extract their DNA. Then we submit it to the biotech center or some other sequencing center, and they amplify or make more copies of the 16S gene, and then they sequence it. And then what we get back is a lot like this, where each one of these is a read, a DNA read, and then it's our job to use computational tools to figure out, who is what. So we do that by comparing what's in our sample to a database. And then we can identify who's there and in what relative concentration, what abundance they're there. And so, what we found when we looked at the population of ammonia oxidizing nitrous ammonium is that if you look at the top graph, there's one organism, so each one of these little de novo 56 is called an operational taxonomic unit. So it's one of those, set of puzzle pieces that match a certain organism at the genus level. So that, at the full scale, was relatively, constant across all 500 days of operation. And we took samples at the same time from both plants. And then in the pilot plant, we saw that guy was there to start with, but then we start seeing some of these other organisms start to become more abundant. So then we can explain some of the adaptation to the low oxygen that we observed in a change in the microbial community as well, or the population of these microorganisms that are removing oxygen. But that's about as far as we can get with that level of sequencing. So I talk about more in-depth details. Ultimately, for this project, we found that these slow oxygen changes were a good strategy for reducing a high oxygen system to low oxygen. And then we also saw that we were able to achieve good total nitrogen and phosphorus removal in the system. And we found that the ammonia oxidizing community had a greater affinity for oxygen. And that was represented either through the flux size, the microcolony sizes decreasing, able to access the oxygen better, and through a change in the microbial community, the abundant ammonia oxidizing population. And then, if you're curious about the nitrite oxidizing bacteria, the ones that complete that second step, they were also enriched at low DO. The reason why I point this out is because it was believed that ammonia oxidizing bacteria would outcompete those nitrite oxidizers for oxygen, and then they might not be able to survive in these low oxygen environments, but we actually found that they were highly efficient at utilizing the oxygen as well. Okay. All right, everybody with me? Okay, we're about to get a little more intense. So in 2015, in the middle of me running this plant, there was this huge discovery, and Dan called Pame and I into his office and was like, you guys, we have to design something to detect these coma mucks. It's gonna be the next big thing. We gotta get a paper published. But we took our time because it was like a side project for us. And then ultimately, you'll see what happens, but we were able to accomplish this, but we weren't the first, unfortunately. So what are coma mucks again? Well, if we go back to ammonia being oxidized to nitrate, for over a century, it was believed that this was done by two different organisms, two sets of distinct organisms, the ammonia oxidizers, like nitrous ammonium, and the nitrite oxidizers, like nitrospira, nitrococcus, and nitrobacter. And they worked together to do this, so that's what was believed. I think prior to 2015, it was hypothesized that there should be one organism that can do both steps because it was energetically favorable. It generates energy for bacteria, so why aren't there any bacteria that do this? And so in 2015, two separate groups in Europe actually discovered that they had one organism belonging to the nitrospira group that actually had the capability of completing both steps. So complete ammonia oxidation. Coma mucks is the acronym that we all love using acronyms. So coma mucks is what they're called. So this section of my presentation I'm going to go through more quickly, but essentially what we did is we took one gene, the AMOA gene, and we looked up the gene in these coma mucks, and then we looked up this same gene in other organisms that are able to oxidize ammonia. And we built a database. And then from this database, we made a phylogenetic tree or a family tree to help us decide, like, what were we going to target with the primers that we designed. And basically from that, we were not able to find one primer set that could target everything. So, because we want to be highly specific in what we target, we don't want to, amplify or count other things that we're not looking to count. So, we ended up designing a separate primer set for each of the 3 Nitrospira Chlomomox bacteria you see up there. So we had 3 sets of primers. And these primers essentially bind to the DNA that we're targeting and allow us to make more copies of it, kind of like crime scene investigators when they want to detect, a DNA. They run it through PCR so they can make more copies, so they can actually see what's there, observe what's there. And through quantitative polymerase chain reaction, or PCR, we're able to back calculate how much we had to start with. And that's valuable because it can tell us, how many of this microorganism is in the system. It's a process to quantify specific bacteria. So, like I said, red arrows are our primers that we designed. We weren't the first, so there were these green arrows over here that also were designed to do a similar job of detecting these Chlomomox. And because I wasn't the first, I got to pay for it because I had to do more work. And I had to compare my primers to other previously published primers. And ours were different because we targeted the 3 different organisms separately, while the others targeted the whole group, which is what we were not able to find a consensus sequence for, but other people designed primers that with specific, I guess you could say, areas that would allow it to be able to bind to many different combinations of things. We call those degenerate bases. And what that means is it's a little less specific, and it has the potential of binding to things it's not supposed to. So I spent a lot of time in this hood, and these 384 well plates, you guys. So many of them, pipetting so many times, and I literally, it was a headache, but I actually really like qPCR because it's one of those processes where you can get results really quickly. So I spend the morning filling this thing, and then in the afternoon I have my results. So it's pretty cool in that sense. So in each one of these little tiny holes, we mix our DNA sample with the primers that I talk about, extra nucleotides, a Taq polymerase that helps make more copies of the DNA, and we put it all together, and we're able to change the temperature of the process to allow us to make 1 copy into 2, 2 copies into 4, until we generate enough copies that we can actually detect it. And in quantitative PCR, we often use a dye, so that the more copies that are made, the brighter it gets. And then it takes a picture, and we're able to convert that into a concentration. So the more that you start with, the even brighter that it gets. So that's how we can back calculate what we have. Okay. First of all, when I compared our set of primers to the previously published set, we found that ours were more specific, as they were designed to be. You can see that by the peaks that all line up on that line. And we found that the other primers were actually giving us a lot of false positives. So that was a problem. And a problem that we talk about in this paper in great detail. But the take-home message from this, which ultimately was our goal, was, are these Chlomomox in Lodio systems? So we compared multiple samples from different bioreactors. We took samples from LSBR, which is the bioreactor that was Pame Kamekos, PSBR, which was Brian Owen's reactor, PCF, which was the continuous flow reactor that I talked about in the first chapter, TRA, which was Trinity River Authority, which was a full-scale plant working with low oxygen in Texas, and NS is 9 Springs here in Madison. That was the only one that had high oxygen. So 9 Springs, we see it was below that dotted line, which was our detection limit. So Chlomomox were not detected above the detection limit in 9 Springs biomass, but it was actually the highest in these 2 bioreactors here, these sequencing batch reactors. And one of the things that those two had in common was that they operated at low oxygen, but they also had a really long solid retention time of 80 days for one, which means that they weren't really wasting from the bioreactor that much to give microorganisms more time to grow. I wish I had some water. Thanks, Mom. So what we found was that homomox belonging to the nitrospira nitrosa group were abundant. We're the ones that were abundant in these low oxygen systems. Oh, thanks. Oh, gosh, you here. So in general, overall, okay, we were able to detect homomox belonging to nitrospira nitrosa in a majority of the bioreactors operated with low oxygen, but they were most abundant in the reactors that had a really long solid retention time. And yeah, take-home message, nitrosa was the one that we detected. We didn't detect the other two. The other primers that we tried, we couldn't get positive results from. They were amplifying a lot of different things, so we weren't able to use them or get any valuable results, unfortunately, which was something that other researchers, when I presented this and at a conference, they also came to me saying like, oh, good, new primers, because these other ones are amplifying a lot of different things. Okay, now we transition. I hope everybody's with me because we're gonna get even more intense into the biology. So, chapter 3, one that Dan didn't read. Yet. I spent a lot of time with this chapter, and one of the things that I wish I did a little bit differently was that I wasn't so intimidated by the microbiology and the bioinformatics, and I kind of didn't dive into this data set as quickly as I wish I would have, because I found some pretty cool stuff when I finally did decide, like, I really have to do this whether or not I'm comfortable with it. So I learned a lot. I read a lot, which is why, it took me quite a bit of time, but I'm pretty happy. I know that this chapter could be better because, and we'll go through several revisions before, obviously. But I'm happy to do it. I feel like the first part of the challenge is just getting something written, and then it's fun to kind of go back and make it better. So this chapter focuses on the key microbes and their gene regulation under alternating low oxygen and anoxic conditions. And this was done in a different bioreactor. So the story of this is, after 3 years, I was like, damn, I can't do this pilot plant anymore. It's taking over my life. I can't go home on the holidays because all my undergrads leave and I have to be here to take care of it. So Dan had some new students coming in, so they took over the pilot plant. And then at the same time that this was going on, one of our other past students, Brian Owen, had been running a bioreactor in the lab that was seeded with Animox from Hampton Roads treatment plant in Virginia. Yeah. And that's a full-scale system. So they sent us the biomass, and Brian started this reactor, and he was running it, and then he was graduating because he was a master's student, so after 2 years. And so Dan takes me into his office, and he's like, so Natalie, do you want to take over this reactor? And I think you were expecting me to say no, but I was like, sure. Also because I needed funding, so that helped too. But anyway, I'm really glad I did it because this is a really exciting process that is being implemented worldwide, and I'll explain why. Okay, back to the diagram of 9 Springs. So, like I said, one of the goals is to make more energy than we use. And one of the ways that 9 Springs is looking at doing that is by adding food waste to their digesters. So the digesters take solids from that mainstream process I talk about, which is full of organic carbon and the bacteria, and it basically heats it up, and there's a lot of biological processes that Matt Side from Nine Springs knows way more about than I do. But ultimately it produces methane, biogas, which can be used for heat and energy, heat and power production for the plant. And by adding food waste, you can make more of that. That's what's reported. However, here in the city of Fond du Lac here in Wisconsin, they do this food waste co-digestion. While it does produce more biogas, it also creates more ammonia to be treated in the mainstream plant, which means more oxygen is needed to treat it. So there's definitely a trade-off. So what's often used in what we call these side stream processes like this is you can use biological processes here as well to treat the nutrients before it goes back to the mainstream so that you can reduce your energy requirements. And the way that we do that is through what we call a partial nitration anammox system, which uses those anammox bacteria that I talked about earlier that work together with ammonia oxidizing bacteria to convert ammonia all the way to nitrogen gas. So we do need a little bit of oxygen for these ammonia oxidizing bacteria to complete their process, but then they make nitrite, and then the anammox take the nitrite and the ammonia and convert it directly to nitrogen gas without requiring any organic carbon. Keeping in mind that organic carbon is something valuable that can be used to create energy. So that's why this is being implemented worldwide in many side stream processes at full scale, and then they're really wanting to implement this in the mainstream process. But one of the reasons that this is difficult is because these guys, the anammox bacteria, they like very specific environments. So they really like warm temperatures between 30 and 35 degrees Celsius in order for them to grow. And they're also very slow growers. So we can't enrich for them in the way that we typically operate these biological nutrient removal systems with a solid retention time of 10 days. They need more time in there to grow and become enriched. So there are these bioreactors called partial nitration anammox systems, like this one. And this is, I was happy to take over this reactor because it's a lot smaller than the pilot. And the reactor on the top left, we controlled the oxygen. We did not control pH, we just monitored it. And we were able to heat the reactor with a hot water that we had recirculating around the tank. So we were able to keep it between that 30 and 35 degrees Celsius using that. So this reactor operated like a sequencing batch reactor. And what that means is that everything happens in one system and we run in cycles. So in this particular system, our cycles were 8 hours long. And we ran it with alternating aerobic and anoxic, and particularly low oxygen aerobic. So we kept that aerobic period low oxygen, and it would be we would add oxygen for 10 minutes, and then we shut the oxygen off for 20, and it would repeat oxic and oxic, oxic and oxic for 7.5 hours actually. During the first 4 hours, we would fill the bioreactor with the water that we wanted to be treated, which is what was, if we go back, it was the reject water from this struvite harvesting process. And this is a pretty cool process that Nine Springs implemented recently to create a high quality fertilizer. So it recovers a lot of those nutrients, nitrogen and phosphorus, magnesium, and it puts it all together in this little pellet that can be applied as fertilizer, and they sell that product. But there's still a lot of ammonia left over, and that's why we wanted to try to see if we could treat that with this system. Okay, so we went to the plant every week. Actually, my undergrad did. and collected full-scale reject water from that Sure-Right harvesting process, and we bring it to the lab. After I took over the reactor, I learned that there were biological processes happening in here as well, so we needed to refrigerate it. So I remember talking to Jackie, our lab manager, about this, saying, Jackie, what can we do? And so the option was either to buy a small refrigerator and like cut holes in it, or to buy a kegerator, which is already designed for beer. So I'm sure that looked really funny on the invoice that our lab was ordering a kegerator. But we were using it to refrigerate this feed. All right. So now if we look at one of those 8-hour cycles that I was talking about for the bioreactor, we can see the changes in the chemical composition. So at the top, we have COD, which is a proxy for our organic carbon, and in this case, we filtered it so that we could measure what fraction of that was soluble COD. We also, and this was, the COD concentration from this particular day was about 70 milligrams per liter. On the right, you can see nitrite and nitrate concentrations from 0 to 15. So there's two different scales here. The yellow is the amount of ammonia coming into the reactor, and you can see that that's increasing over the first 4 hours of the reactor operation because that's when we were filling it. But some of that was reacting at the same time because we were still operating with these really short oxic and anoxic cycles. So that's where you see these little spikes in the orange, which is nitrite. So the ammonia is getting converted to nitrite, and we can detect that. And then nitrate didn't accumulate on this day. On some days it did, which indicated that we had some nitrite oxidizing bacteria, which are not ideal because they compete with anammox for the nitrite. So in an ideal reactor, we get rid of those organisms and only have ammonia oxidizers and anammox. But ideal is not reality. So before I move on to all the more genomic topics, I want to just overview of how a bacterial cell works for those of you who may not be familiar. So I like to use this analogy that a bacterial cell operates a lot like a factory. So this is how I explain it to my parents. So DNA is the instruction manual. When we look at the DNA, it tells us what the potential of this factory is. What can this factory make? Just from the instruction manual. But that's it. The RNA is when a worker goes and reads the manual and translates it into something that can be understood by other processes or workers. Okay. Proteins and enzymes are the product of the workers reading these instructions and translating them and making a product. Okay? And the proteins and enzymes are the machines that do work in the cell. A regulator is the manager that tells the workers when to make an item and when to make an item and it basically tells it when to make an item and when to stop making that item. And then a global regulator is the CEO that can control many workers and decide when they all are supposed to make an item and stop making that item. Okay. In our system, we were interested in that CEO, the global regulator. And this global regulator is called FNR, which stands for fumarate and nitrate reductase. And I give all the credit to the FNR work to Kevin Myers in the back, who his work focused on FNR in E. coli. So he helped me look for these FNR binding sites, this regulator, in my microbiome. And the reason why we care about this is because FNR is required for the expression in energy generating pathways, specifically in our case, in those that cycle that organic nitrogen, and it's an oxygen responsive regulator. So in E. coli, is responsible or required for the switch from aerobic to anaerobic metabolism, which in our reactor, that's how we're working, oxic and oxic. So we wanted to see, is this playing a role in how our microbes adapt to these quickly changing environments? Okay. Water. We're getting there, I promise. So to start, we first have to sequence and get genomes for what's in our reactor. So we decided we took samples from 4 different days of the biomass where we had variable nitrogen removal, and then 16S sequencing that we used. We were able to find that we had, a denitrifying microorganism, an ammonia oxidizer, a nitrite oxidizer, and we also found our animox. So even though our reactor was not red, like you would expect an animox reactor to be, it was still there, but it was at really low concentrations, much lower than what you would expect to find in an animox reactor. And we were able to achieve almost 90% nitrogen removal, so that's where we were kind of scratching our heads like, okay, then what's going on? If animox is so low, why is this process so efficient? So that's what fueled the motivation to do this whole genome sequencing. So if we go back to this little picture, to do this, we, again, take a bacteria sample, beat them up, and take their DNA. And then, instead of just sequencing one small part of each microbe, we actually chop up the DNA of everything and sequence everything, which gives us this. So, in my case, I generated, after I submitted the samples to the biotech center and they gave me the data back, I had 200 million pieces of DNA. And so in order to process that amount of data, you have to use computational tools and high throughput computers. And so I had to teach myself coding and bioinformatics, and Garrett Suen is responsible for my very first bioinformatics class, which was well before I did any of this, so that was really helpful, because I was able to go back to my notes and look at that. And so I compare that to like the world's biggest puzzle. This is a real thing, and it's like $1,000. So how do we begin to work with data like this? Well, like I said, we use computational tools. So we start off by assembling these reads. So we take pieces that are overlapping, and we can put them together, just like you would a puzzle. Then, and I use metaspades in this case, and then we can start to put similar looking pieces together, and we call that binning. And so I use a wonderful tool called Ambio. So for any of you that are working with a data set like this, I highly recommend it. Because Ambio lets you, it automatically does this, but then it lets you go in and manually curate each one of your bits, or what we call metagenome assembled genomes. And so this is what I did to parse out, organism A, organism B, organism C from the bioreactor. But this only tells us who's there and what their potential activity is in the system. So we have to go one step further. And we also extracted the RNA from this. So what this tells us is not only who's there, but who's active. So we took 4 samples across one of these little 30-minute, actually 2 of these 30-minute periods, where it's oxic for the first 10 minutes, or low DO, so we're adding oxygen. And then for the remaining 20 minutes, there's no oxygen. And then we see that nitrite accumulates in the orange, and then there's nitrate as well. So we took 4 samples, and we flash froze them because RNA is unstable. And then we extracted the RNA, and then we are able to match up those reads to our genomes, and then count how many there are and get an idea of their gene expression. So this is where I spent a couple weeks in Trina's lab, extracting the RNA with Matt and Pancho. And this was a really anxiety-driven process because once you get going, your sample kind of disappears. You can't see it anymore. and you don't know if you did it right until the very end. So luckily, I was working with some people that knew what they were doing, and we did end up with the RNA that we were looking for. And I remember telling Dan that, oh man, that was tough. And Dan was like, that's not the hard part. The hard part is processing the data. So yeah, you were right again. So, but yeah, I did it. And the metagenome assembled genomes from the metagenomic sequencing and the RNA sequencing together, we were able to use the data to get an idea of relative abundance and relative expression. So with the whole genome sequencing, I was able to assemble about 77 highly complete genomes. That's a lot to work with. Many people will just focus on one. So, but because this was a microbiome where there's multiple players involved in nitrogen cycling, we needed to look at more than one. So luckily, the top 4 most active microbes were 1, a chlorobi, which is often involved in denitrification. 2 are Anammox, which surprisingly, even though it was low in abundance, it was the 2nd most active microorganism in the bioreactor. 3rd is our ammonia oxidizer, and 4th is our nitrite oxidizer. So that was pretty cool. So then I decided to focus on 4, which I realized when I was writing chapter 4 that was a lot still. But here we are. So to begin to kind of put each of these genomes into context, figure out who they're related to, we have to download genomes from the internet and from databases, and then we build a phylogenetic tree, which is a lot like a family tree. So it tells us who's related to who on an evolutionary context. And then I looked at all 4. So CHB, it did correlate with the chlorobi OLB4, which originates from a Anammox system in the Netherlands, which is pretty cool. And ended up that they were actually nearly identical. AOB was nearly identical to Nitrosomonas europaea that I used other tests to determine. AMX was similar to a Brocadia fulvita and is actually almost identical to Chris's Anammox from his Anammox community interactions paper, which was pretty cool. And then NOB down here clustered with this Nitrospira group, and I thought it was going to be a Nitrospira diffluvi, but when I did further testing, it actually ended up being nearly identical to a different NOB belonging to the same reactor in the Netherlands, and it was not considered a Nitrospira diffluvi strain. So to further compare the genetic content between related genomes, I wanted to know maybe what genes were unique to the organisms in my system and which ones were shared across other related organisms. So I generated 4 metapan genomes and using Ambio, great tool. And what this shows us is each concentric circle represents the genetic content of one organism. And if it's shaded, it means it has it. And if there's multiple regions that are shaded that line up, it means they all have that region. And I'm able to get a database from that and look at what was shared and what was unique and stuff like that. So I was able to also, in these regions, create different categories and use that to help my analysis. So this is the one for the Nitrosomonas organisms, and I have to laugh because when I was working on this, my husband could not get over how similar it looks to the Death Star. And so that was the purpose of this, was not to be the Death Star, but to help me figure out how my organisms were unique. So then the RNA sequencing finally came in, and we were able to look at, and I went through each of my 4 genomes with a fine-tooth comb. I annotated it probably in 10 different ways, which I realized was even more confusing because these different databases will tell you something different. So ultimately, I used a database called NCycDB, which was built and manually curated to contain all of the genes related to nitrogen cycling. So, and then it would tell me, how similar they were, and I used that to help me find these genes related to inorganic nitrogen cycling. So this is a really good table. I have to thank my husband, John, because I had him sitting next to me, telling me, okay, is it, I was reading off these genes, and then we were looking for it, and I had my mom helping me, trying to, we had big Post-it notes on the wall. I sent pictures to Dan where we were, cutting out pieces of paper and saying, okay, this one has this gene, this one has this gene. We're trying to keep track of everything. It was kind of crazy, but ultimately what I found was that this gene profiling showed that each of my 4 microorganisms, they did contain what we expected as far as gene content. But they also showed that they potentially had functions that made them really versatile in different environments, and that was really interesting. For example, with aerobic respiration, I looked for some of these terminal cytochrome C oxidases and found that my denitrifier was not an obligate anaerobe, but it also had the ability to respire oxygen, which I think is similar to what Chris found as well. And I also found that my Animox also had the genes encoding for these aerobic respiration pathways. So I also found other papers that said that they do contain these cytochrome C oxidases, but they don't necessarily understand if that's because they're actually using the oxygen for growth or if they're doing this to detoxify their cells, because they are supposed to be obligate anaerobes. So I not only looked at that, but then we're getting close to the end. I evaluated also the expression profile, so how these gene expression was changing over time, of 350 genes related to inorganic nitrogen cycling. So that's why I put my family to work, and they were helping me try to keep everything straight. So ultimately, I was able to group these using a computational model that puts similar gene expression changes into a group. And I did all of the 4 organisms separately so that I could compare how the gene expression of Animox compared to the gene expression of some other organisms, and then which genes belong in which group. So yeah, this is why I tacked this one. It took me a long time. Not only that, we also used the data from Kevin's work on did these genes contain this FNR regulator? That's an oxygen responsive regulator, and which ones had it, and in which groups did they belong? And then which genes contain the FNR regulator and in which groups? And I talk about all of that extensively in that last chapter. So here's one example. So this particular group, group 4, contains genes from fluorob, the AOB, and NOB, and Animox. And it shows the expression changes. The blue line is the mean change in expression, and then that shaded region around it is the plus or minus 2 standard deviations. So what I found by clustering these genes from each individual organism separately is that they had some expression profiles that were highly similar. So AOB and CHB, NOB is a little bit different because it goes down in that anaerobic period. The shaded region, I should point out, is the aerobic samples. And then Animox. So you can see here that all of these are upregulated during that aerobic period. And I found genes related to aerobic respiration in all 4, nitrite reduction to nitric oxide, and nitrous oxide reduction to nitrogen gas. And a majority of these genes in this group in particular contained a predicted FNR binding site. So what this tells me, we'll go through the general summary, but what this implies and needs further investigation, and I look forward to hearing what my committee has to say to make this chapter even better and stronger. But what this may imply is that FNR may in fact be a global regulator in this microbiome, regulating these genes involved in inorganic nitrogen cycling. Okay, so ultimately from this study, we were able to extract and assemble highly complete genomes belonging to these 4 groups that are commonly found in anammox systems. So 3 out of the 4 of these were also found in an anammox bioreactor in the Netherlands. Interestingly enough, I started reading about this bioreactor enough to find that it also treats the reject water post-struvite harvesting. So it has that in common. And both bioreactors, they are operated fairly similarly in the Cannon type process. But the anammox system that I found was not similar to that bioreactor. It was actually more similar to Chris's study, which originates from a bioreactor in Utah, which operates differently. So I found that pretty interesting. Okay, the gene expression profiles, they did show oxygen-responsive changes in transcript abundance. And I found the similarities between the different expression profiles quite striking between all 4 organisms. And the presence of this predicted FNR binding site upstream of the transcription start site may indicate that it does indeed control the turning on and off of these nitrogen cycling genes in these switching environments. Okay, so overall, tying everything together, we see that low oxygen, nitrogen, and phosphorus removal was successful at the pilot scale. And this is really important because it can save a lot of money and energy just by reducing the oxygen and not having to upgrade anything else in the plant. Comomox may indeed contribute to low oxygen nitrogen transformations, but it appears to be more important in systems that have a longer solid retention time. I was surprised to find that anammox, and I guess not surprised because a lot of the papers seem to point this out, that anammox and other nitrogen cycling organisms in those type of reactors are highly similar, despite geographical region and sometimes configuration. And then the activity of anammox and other nitrogen cycling organisms may, in fact, be regulated by this global FNR. Okay, knowledge, we made it. So I'd like to acknowledge, Madison Metropolitan Sewage District. Thank you so much for mentoring me and helping me get the pilot plant up and running. Pauline, Steve Reeser, Alan Grooms, and Matt Seig. And also thank you for being here today. It means a lot. Jacobs and Trinity River Authority, they helped collaborate on the Comomox project and getting these samples, and that was really great as well. Of course, my advisor, Dan, you've been great. And I tell students coming in that your advisor can make or break your experience in grad school, and you definitely made mine. So you're an excellent advisor. You give advice when it's needed, but you also let us be independent, and that was really important to my growth. So you were there for me whenever I needed you, and you let me go off and figure stuff on my own as well. So that was really key. Jackie, thank you so much. You've been awesome. I'm really gonna miss you. Jackie's our lab manager. She helped train me on equipment, and then at the end, I helped train you on the microscope, which was great. But you were always there to talk to and have great conversations and help get my undergrads up and going. And I mean, you make the lab run. So I thank you for that. And of course, my current and previous undergraduates, I'm really proud of them. Several of them have decided to go into grad school and have emailed me saying that I really helped them appreciate research, and so that means a lot to me. And even one sent me a gift recently, just thanking me for being a reference for her for a job search, and They've been really great. I also want to thank my co-authors on my first 2 publications. Thank you for helping put those together and making it something that was accepted. and then the last paper, that side, I'm sending you a draft. It's it's finally written, so that'll be heading your way soon. For that last paper, my committee members, thank you so much for participating on my committee, for advising me during my prelim that I should only get 1 PhD and not 4, because I learned that I should not propose all the things, because really I am terrible at estimating how long things take. So hopefully I will learn someday how to be better at that. And I recognize this, that's the first step. My parents, thank you so much for being here, for your support all this time, and letting me convince you to let me stay in Wisconsin for 4 more years. My Wisconsin parents, Jim and Peggy, thank you so much for coming to this, to my graduation. It means a lot that you're here. My husband, John, you were incredible. You were there through every tear, every smile, every discovery, every failure, everything. You saw it all. So, and you fed me. Thank you for cooking. Yeah, really though. Especially during times like these, like especially in this last push and the prelim and falls and everything, you were there to feed me and provide me with sustenance. I'd like to thank my acquired siblings from John's side. They have been wonderful as well. And I have a video that I put together. We don't have to watch the whole thing because it's basically 1-second snippets from the last 6 years that are all combined into one video. I didn't do this manually, I have an app called 1 second every day. But I thought it was pretty cool to reminisce on some of these memories. So it's 8 minutes long. I guess that's as much as I could condense 6 years. So we definitely don't have to watch it all. You guys can be the judge of that. It's been an hour and 20 minutes. So I'll let my committee tell me when to cut it off. But here we go.
Don't be a chicken.
Oh, swirly rainbow Jack.
Road train.
That's the most unhelpful thing I've had ever. Okay, I think I'm gonna see stuff. We're in 2018 now. Okay. So with that, I am open to any questions.
Okay, it's open for the attendance for questions. After questions from you all, we'll go into closed session. So please ask your questions.
Okay.
So you did a lot of interesting work on the FNR regulator and on how it regulates transcription in your system. I'm wondering if you think that with respect to oxygen, if your system's more regulated at the transcriptional level or post-transcriptional, maybe post-translation.
Yeah, so I think that that's something that definitely requires further investigation because it's definitely a possibility that there's post-transcriptional regulation. What I've read is that often bacteria are regulated at the transcriptional level. So that's what leads me to believe that FNR could, in fact, be significant to these systems, but it doesn't, I mean, there definitely could be other things in play, and just because something is transcribed does not mean that it's being made into the protein that we're talking about. It just indicates potential.
Yeah, and I guess that's where more of my question is coming from. Do you think it makes sense that they're resynthesizing their proteins every small cycle of air pulses that you're giving them?
Well, I think in some of the clusters that you didn't see here, in some cases the gene expression, I guess in some of these the gene expression didn't change in some of these nitrogen cycling genes, which means that they could be keeping them constantly available. But in some cases, I saw that the genes were expressed prior to where they would be used. So, like in an anaerobe, like for example, there were some genes that are used aerobically that were being upregulated anaerobically, almost as a strategy to be ready, I know, lack of a better way, for that oxygen availability in order to utilize those enzymes. But yeah, I think that to answer that question, there would require some additional maybe proteomics or something like that. But yeah, does that answer your question?
Yeah.
Okay. One of you, Matt?
So you saw the flock size shrink down in your pilot reactor. What would you suggest as some methods to test for change in sludge settlability? I know you didn't really look at that a whole lot.
I did actually, every time we went to the pilot, we did do a settlability test, the 30-minute settling test, and we recorded that. The settlability of that pilot was not as good as the full scale. Now, one of the things, of course, that clarifier was not optimal either. It had a very small surface area, and so how that impacted the flock formation, I don't know. And then methods for improving, determining settlability, or is that what you were saying?
Yeah, just trying to get a sense for how would you, what tests would you use to characterize the settlability of the solids? You know, if we needed bigger clarifiers or more clarifiers. Or, you know,
That's a really-
SVI is one test, but what else could we?
What else could we do? Ooh, I haven't thought about that topic in a while. I honestly, I don't think I can answer that at this time. Other than what we did with the settling tests, I can't think of anything off the top of my head at the moment. But I would love to discuss it with you. What ideas you have. I assume you know the answer to that.
I don't.
Steve?
Oh, yeah, first I was just gonna apologize for the condition of my.
It was a learning experience though. Like I took a lot from that.
Yeah. I was just wondering as far as chromomox, evidently they don't develop in the analox reactor.
I did not detect, no, my nitrospira was not a chromomox.
So they would a problem if they did.
I don't know what the status is of other analox systems, whether or not it's there, because that could be, that is something that is talked about. How do these chromomox impact these analox bioreactors, because they go all the way to nitrate, but they may, there may be a link between them and denitrifiers that recycle that back to nitrite for analox. So it may not be detrimental to an analox system, as long as there's other microbial interactions that facilitate that, that little loop, nitrite loop.
Are there other good situations for using chromomox?
I would say a good situation, well, if they are efficient at utilizing low oxygen, and if there's no total nitrogen requirement, although denitrifiers could potentially denitrify the nitrate, but then you have the carbon issue, right? So I'm not sure. I haven't thought about in like what particular, other than thinking about them from a low DO standpoint and just understanding their presence in those systems, I haven't thought about where they could be used beneficially, but I'm sure engineers can come up with something. Yeah.
Maybe somebody with a PhD would.
Yeah. The next PhD. Yeah, Paul?
Natalie, one of the practical concerns we had with the low DO reactors was how quickly can they recover after an upset? Be able to do any work in that area?
I didn't, well, I guess in one sense, indirectly, I remember losing biomass particularly and freaking out, telling Dan, like, oh my God, I'm gonna have to start all over. And he's like, it'll come back. And it did. The recovery of phosphorus removal, I mean, I rarely lost phosphorus removing capabilities, even during upsets. With ammonia, if the upset happened in the wintertime at low temperatures, it took quite a bit longer to recover completely. But I would say usually within, I mean, this is too long for a full-scale plant, a month. But the students that came after me started looking at why we were losing, ammonia removal during the winter time and what strategies could we use to combat that. And I think one of the recommendations was to maybe increase the oxygen a little bit in the wintertime, 1, and 2, to increase the solids retention time to kind of combat that slow growth at lower temperatures. I don't know if that answers your question, but okay.
Any other questions? Okay, let's thank Natalie once again.
Yeah, I think so. Hey, John, you can stop.
Oh, yeah.